So this session is called Genetics Made Simple. This is intended to be kind of an introductory session about genetic testing, how it can benefit an individual who has an epilepsy syndrome like Lennox-Gastaut, and just walking you through how the testing works, what are the benefits, what are some of the things to look out for, how you access testing. So there's two of us presenting today. I'm Beth Scheidley. I'm a genetic counselor at Boston Children's Hospital. And with me today is my colleague Jillian Frinsing, also a genetic counselor. So we'll be tag teaming a little bit throughout the course of the day, but I'm really curious before we get started, how many of you are parents or caregivers of an individual with LGS? Any siblings? Any non-family members? All right. And just a show of hands, I'm wondering how many of you have family members who've gone through the genetic testing process. Maybe about half, 2/3. And how many of you, as a result of that process, have arrived at a definitive genetic diagnosis for your family member? So not very many out of those of you who've started down the road of genetic testing. How many of you have an uncertain diagnosis that you're kind of waiting on? And how many of you have just had negative test results? Some overlap there as well. All right, well, I think we're hoping to kind of walk you through the current state of things. There's a lot of change. This is a really rapidly developing area in terms of both evaluation and research. So I'm just going to skip real quick to show you our agenda. So we're going to start with a little brief history about the genetics of epilepsy, what we've learned over the last 50 or 60 years. Then we're going to shift to some fundamental concepts in genetics that I think will help you to understand the commonly used genetic tests that we offer to families that we see at Boston Children's. And then how to interpret genetic test results, how to figure out which genetic test is best, how we do that, how you might think about it if your loved one has already had some genetic testing, and then we'll walk you through interpreting some actual test reports, and then we'll finish up talking about how to access testing and some other special considerations in testing that just didn't really fit into any of these other categories. But I want to go back real quick and just ask, just by way of introducing the idea of genetics, how many of you can roll your tongue like the little boy on the left? Oh my gosh, lots of you. And Yvonne can do it. Lovely. Yvonne is our genetic counseling assistant. Why don't you stand up there for a second? Yvonne is planning to go to graduate school to become a genetic counselor, and that is one of the entry criteria. So, and you can as well. Lovely. Well, it turns out there's actually a genetics responsible behind who can curl their tongue and who cannot. I definitely cannot. My husband can actually double roll his tongue. I'm not really sure what that means, but how about look around at your friends and neighbors to see whether their earlobes are attached or detached from their head. You're definitely going to see some differences. That's actually due to genetics as well. So my point just being here, there are lots and lots of observable traits that we think about every day that are just common traits. And behind those traits are genes and chromosomes and genetic changes that may be common in the general population or might be more rare, especially when we're thinking about genetic epilepsies. So I want to take you back through a brief history of the genetics of epilepsy and just sort of theories about causation. I'm sure you are all familiar with some of the centuries-old theories involving spirits or demons, which thankfully, taking the place of that was more scientific medical knowledge about what some of the causes of epilepsy might be. But even, back in the 1970s, there was this large category called idiopathic, which basically meant we don't really know, right? But it turns out a big chunk of that idiopathic part of the pie are genetic changes. And that's really what we've started to learn in the last just 10 to 15 years. So, back in 1994, when I graduated from my genetic counseling graduate program, so that's me in the little cap and gown, that was before any epilepsy genes had been identified. So when I finished my training, we knew absolutely nothing about the genetics of epilepsy, and I can tell you that one of the very first families that I saw as a student was two parents expecting their first baby. Mom had epilepsy, dad had some cognitive issues, and their primary concern and the reason for the visit was the medications that mom was using during pregnancy and what impact that might have on the developing baby. But it came up during the session, what's the likelihood the baby's going to have epilepsy? And we really had nothing to go on back then other than just kind of family studies of, okay, if you have a first degree relative, the likelihood that you might have epilepsy or your child might have epilepsy is X, Y, or Z. But we knew nothing about the genes. In the 10 years or so that followed, there were a handful of single genes that were identified that play a role in epilepsy. So you probably heard about some of the channelopathy genes like SCN1A or SCN1B. But then there was a really long lag time where there was a lot of genetic research going on and just not much that was being identified, not much was being learned. This particular visual comes from a paper from Dr. Ingo Helbig and Dr. Sam Berkovic, just demonstrating a really kind of lengthy period of time when we weren't learning a whole lot. Lots and lots of studies. But around 2011, 2012, there was a new genetic technology that came on the scene called next generation sequencing, which enabled us to sequence the human genome rapidly at low cost. And that, coupled with the completion of the Human Genome Project, which was at the tail end of last century, maybe we're in 1999, 2000, really paved the way for rapid gene discovery. So about 10 years or so ago, when I gave a talk like this is a slide that I showed where, the blue bubble is back in the 1990s, early 2000s, and then the pink section, which you can see is really exponential growth in terms of new gene discovery. Lots of new genes identified because of new testing technology that came on the market from commercial labs, like gene panels, which Jillian will talk about in a few minutes. So all of a sudden, we started learning a whole lot more about the genes involved in epilepsy, and in particular in early onset epilepsy syndromes, things like Dravet syndrome or LGS. Now here's where we are at the end of 2023. And obviously you really can't read anything on that slide, right? The point is just look at that. I mean, look at how many genes. There's estimated to be over 1,000 different genes associated with epilepsy at this point in time. So the discovery is rapid, and that's a double-edged sword, right? Because we're able to make genetic diagnoses much more readily. But that also means with new gene discovery, we don't always necessarily know a lot about what those findings mean, right? So we talk about this all the time at Children's. Every week, every couple weeks, we get a test report back from a lab, and it's a gene that was just first reported in the literature 6 months ago, right? It's the first family we've ever seen with an alteration in that particular gene. So there's a lot to learn, but a lot of excitement about how much new information we're getting at a very rapid pace. So that kind of just sort of sets the stage for where we're at in terms of being able to make diagnoses because of all of these new testing technologies, which you'll hear more about. But at the end of the day, the question becomes, why is that even important? And if any of you went to Tracy Dixon Salazar's talks, you heard her talk about, the Pokemon, right? And the different diagnoses, and you got to catch them all, right? Your child has LGS, but your child maybe also has a genetic diagnosis, right? And why is that important? Well, first of all, there are a very small handful of genetic diagnoses that would immediately lead to potentially a change in treatment, either a medication that works better or avoidance of certain medications. And the best example I have of that are sodium channel blockers, which you wouldn't want to use in an individual who has an SCN1A gene-related epilepsy. So in some cases, a genetic test result may have a dramatic impact on what your physician tells you in terms of selection of epilepsy meds. Aside from that, for many families that we meet, especially when their children are a little bit older, their children have been poked and prodded a lot, right? And I don't have a child with epilepsy, but I have a child who's gone through some pretty significant medical issues over the course of his life, and I know what that's like when your child has to have, MRIs or endoscopies or procedures. It's very hard for the child, it's very hard for the parents, and if you can avoid that by having either a test done with a cheek swab or one blood draw and not have to go through all of these other procedures to arrive at a diagnosis, to me that feels like a no-brainer, something that you would want to do. Aside from that, a genetic diagnosis can give you more information about prognosis. So your child has LGS, but your child has LGS due to a specific genetic variant. There may be a whole world of information that you then have access to because of that genetic diagnosis. You have access to that particular community. It might lead to referrals to other specialists because maybe there are things about the prognosis associated with that gene that suggest, maybe there should be monitoring of blood sugar levels, for example, if diabetes might be more likely because of that genetic diagnosis, or maybe you want to just check and make sure there isn't a cardiac defect or a renal defect with an ultrasound. So the referral to specialists can be a really important part of getting that genetic diagnosis. In some instances, unfortunately, we do make rapid early diagnosis. This is usually with really young infants, right, where there isn't a whole lot that we can do, and the family might want to be opting for palliative care, and having that information really helps them to make those decisions more readily and meet with the palliative care team. Finally, if nothing else that I've said really resonates with you, I would say that increasingly, I think we are going to see treatment trials that are based on either the gene or the specific genetic variant. And so having the genetic diagnosis might be the eligibility criteria to get into that trial, right? So there might be a trial where the child must have LGS, but it must be due to a specific gene, for example. So it's kind of like a union card to getting into some research studies down the road. So that's sort of the clinical utility of a genetic test result, but in addition, for some families, even if it doesn't really impact treatment or prognosis right now, they might want to use that information for reproductive planning if they're thinking about having more children. The specific genetic diagnosis can tell you more about the chance that another child might have the same diagnosis. With most genetic epilepsies, the likelihood of recurrence is actually pretty low because most genetic epilepsies arise what we call de novo, or new in the child. But you can't really know that without having the genetic diagnosis. If you have the genetic diagnosis and you decide that you want to have more children, it would make prenatal diagnosis a possibility through amniocentesis, for example. It might also be possible, depending on the diagnosis, to do something called pre-implantation genetic diagnosis, where you do IVF and you have the embryo tested before you implant. And then testing of other family members, if there are siblings who are thinking about having children, or other family members, again, depending on the inheritance pattern. And then really for some families, it's really just, having an answer, maybe being able to put aside the theories that you've had about why your child has epilepsy, or blaming yourself for something you did or didn't do, either before you got pregnant or during the pregnancy. Those things can really loom pretty large. And then finally, just access to support organizations. I know you all have access to LGS, which is fantastic, but some of the gene-specific family organizations could be really helpful as well if you have that particular genetic diagnosis. So I think I'm going to leave it there for now and invite my colleague Jillian to come up and review some just fundamental concepts in genetics and genetic testing options. Are there any burning questions right now? We were mostly going to save for the end, but if there's any burning questions right now before we move on. No? Okay.
Hello, everyone. Can you hear me okay? Okay. So as Beth mentioned, my name's Jillian. I'm also one of the genetic counselors over at Boston Children's. And we're going to go through some fundamental concepts in genetics. I saw that most people had genetic testing done at some point, so hopefully this will sound a little bit familiar. But if there are any questions, we will do a little break after this session, or section. So going back to kind of what we asked in the beginning, if you could either roll your tongue or if your earlobes were detached or attached, those are different traits that we all have, and that's the presentation of those, and we use the word phenotype to describe that. But that is all driven by our genotype, so what is written in our genetic material. So when we see symptoms in an individual, we then say, can we find an underlying genetic explanation for that? That would be the genotype. So you might hear us use these words, and some of the tests really rely on the phenotype or the symptoms that we're seeing in an individual. So this slide was just to show, hair color is also, a trait we might see, eye color, things like that. We're all remarkably similar when we look at the genotype. So 99.9% of our genetic material is identical across individuals. But that 0.1% makes up for thousands of variants. So a very small percentage, but a large number of variants. Most of those variants are just determining those traits like hair color and eye color, but some of them can be in places that we see lead to different symptoms in individuals. And that's where we will start talking about proteins and what make up proteins. So on the left, you can see a typical individual. We're mostly made up of water, and then proteins. And proteins are made up of different building blocks, and they need to link together properly. They need to fold together. That's what we call the amino acids. And then there's some fat and there's some sugars, which we call carbohydrates as well. A note on proteins, this comes up a lot when I talk with families. We're not talking about the protein in our diet. It's just a general term that we use in biology and genetics for anything that has a very specific function in the body. So proteins that we might be talking about are like antibodies, so that would be a very specific function to help us fight an infection. It could be in our blood, so the portions of the blood that help us deliver oxygen, or enzymes, so something that's going to help us break down food into the nutrients to be able to use that for energy. And specifically for our talk today, there are many important proteins in the brain. Most of their jobs is to help regulate what enters and leaves a cell and regulating those electrical activity when we're talking about epilepsy. So some things you might see are receptors, transporters, ion channels, or pumps. So all just different ways to regulate what's coming into a cell. And I know a lot of people said they had genetic testing. Had anyone ever seen a diagram like this or something similar to this as going through part of their genetic testing process? No. Okay.
How many of you had a genetic counselor involved in your testing journey? Many of you are going to have testing.
Yeah.
Yeah, okay.
So at Boston Children's, we typically do consent before and meeting with a genetic counselor, and we will show something like this. The idea is not that you will become an expert and maybe have interest in this, but just so that you know what testing is being done and what we're looking for. So we hope that this will become familiar after we go through it today. So I already mentioned a cell that's all the way on the left-hand side of the screen. That is the basic building block of all living things. So the smallest that we can get, and then they link together to build tissues, to build organs, and to build our body. But every cell in our body contains a copy of our genetic material. So we typically talk on a one-cell level. And our genetic material is in the center there in a structure we call the nucleus. It's typically in the center. And those chromosomes, which are to the right, are the way that we store the genetic material, because we just have so much of it that we need it to fit really nicely inside of the cell. And so chromosomes hold our genes. Genes is the type of testing that we typically do nowadays. We'll go into those most common tests. But a gene is just a word to say it tells us how to make a very specific protein in the body. So every gene is telling us either to make one or multiple proteins. And then that protein again goes on to play a very specific role in the body. And genes are made up of different letters. There's 4 of them: A's, T's, C's, and G's. And their official name is nucleotides. You can see that on the bottom. But those letters need to be in the correct order, and there needs to be the correct number of them, or a protein might not be able to be made in general, or it might not be able to do its job. And that's typically when we see symptoms in an individual. And just to get into a little bit more detail, we talked about how it's made up of letters, but we actually read through them in sets of 3, and each 3 tells us how to make one of those specific building blocks of a protein. So we could actually be missing one of those letters, and then every building block after that is going to be different than expected as well. So different types of changes can lead to things, and we'll go into those in more detail. I think I went through that. And then I've been saying that genes make proteins, but haven't really talked a little bit about how that happens. So the official words are transcription and translation, but we need to take our DNA or genetic material, turn it into RNA. So I know I talked to some families earlier that we're doing RNA studies now. And then we turn that into the protein. And I bring that up because if we actually look at a gene, there are different regions that we can look at to see if we can find an explanation. So on the left-hand light blue side, we have a promoter. That's typically where we're going to start, where the gene begins, and we're going to start reading through. And then in dark blue, we have what are called exons. Those are the portions that go on to make the protein. They hold the information for it. But we have red spaces in between called introns. And as you can see, those get cut out or spliced out in the process. Those are more regulatory, so making sure we include all of the exons or protein coding regions, or telling us how to make the protein. So before we would only look at those exons or dark blue regions, but we're learning that in those spaces we can find changes that are just as important. So everything we talked about so far is typically in the center of the cell called the nucleus, where we get half of the genetic material from our biological mom and half from our biological dad. But we also have mitochondria in the cell. They're a little bit harder to see, but we tried to circle them in blue. Their entire job is to make energy and provide energy for the cell. If they are not functioning, we can see seizures and developmental delays and other symptoms. The main thing is their genetic material is in a circle, and it's completely different. They have their own genetic material not included in the nucleus. It actually only comes from biological mom because it comes in the egg and not the sperm. So this is a separate test that can sometimes be done. So that was the nitty-gritty genetics terms. And after we go through that, we typically try and zoom out a little bit and make it more applicable to something in our everyday lives. So we could think about all of our genetic material that we call the genome like an instruction manual. And then the chapters within it like the chromosomes. So we need all of the chapters to be there, and they need to be in the right order. And then the genes are like the sentences. So we need to be able to read through to understand how to make that bookshelf or dresser. And then we also need all of the words to be there. Those would be like the building blocks of the protein. If one is missing, we might not understand what that sentence is saying anymore. And then even within the word, we talked about how the genes are made up of letters. If it's spelled incorrectly, that could also lead to us misunderstanding what we are supposed to be making, just like the body. So that was a brief overview of genetic concepts to frame genetic tests that we might recommend for individuals with epilepsy or Lennox-Gastaut. Before we dive into that, I want to ask if there's any questions or specific things you've run into in your experience that we could clarify.
Hold on one second and get the microphone.
Sorry, the microphone is
That's
coming.
okay.
Can you clarify when you were talking about just a few minutes ago about
Yep.
the own genetic material, and you said something about it comes through the mother from the egg? Can you explain
Yes.
that?
So I believe it was this part. And I apologize. I thought I was going to be able to like run over and point to the screen, but we're being recorded, so we're up here. So I think I'm going to go run over there.
There is a little pointer, I think, on the top button.
Ooh, okay. So our genetic material that's in every cell of the body that we typically talk about is called nuclear DNA or genetic material. We get half from mom and half from dad, and that's going to be stored here. But then we also have these structures called mitochondria, so they're outside of the nucleus but also in the cell, and they just make energy. And they have their own genetic material, really because they used to live separately of humans, and we decided they were really helpful to make energy, so we took them in. And their genetic material here, we can also look at that, so look for any letter changes or differences. So when the egg and the sperm come together and we get half of the genetic material from mom and dad in the nucleus, we actually only take the mitochondria from the egg and not the sperm. So that's why it comes from biological mom and not from dad in that case. Does that answer your question? Okay. All right. Very good question. Sorry.
Please wait for the microphone.
Just so the people at home can hear.
Is this the name of that test specifically?
Good question. So it's mitochondrial DNAs to separate it from nuclear DNA, and then there is a test that you can do that we'll get into either separately or it can be included with another test that is also looking at the nuclear genetic material.
Okay.
Very good question.
So my daughter was born with Down syndrome, and her neurologist told me that we didn't need to do any genetic testing because she already has a Down syndrome diagnosis. So my question is, would it still be beneficial to do full genetic testing?
Is it okay if I go to the next slide to answer your question, like start going in? Okay. So common genetic tests. Right before I get to you, that'll be the next slide. There is not one specific test that's best for everyone right now in 2024, just because we can look at different things. The nuclear DNA, the mitochondrial DNA, we can look for Down syndrome, we can look for explanations for epilepsy, things like that. So it might be a stepwise process for some people, and sometimes more than one genetic test is actually necessary in the process of genetic testing. And then we always give the caveat that interpretation of genetic testing is very difficult. The lab reports are typically in very complex language. So we recommend talking to someone, particularly the person who ordered it, to talk about what it means in detail, or seeking out a genetic counselor or geneticist. So specifically kind of related to your question, chromosomes, again, just as a refresher, are the tightly condensed form of our genetic material. They look like X's, and they are stored in the nucleus, and we have 23 pairs for a total of 46, and within each pair we got one copy from mom and one copy from dad. And then typical males have one X and one Y, and females typically have two X chromosomes. So that's what makes us different on a genetic basis. This is one type of genetic test that you can do that's called a karyotype, and it essentially takes a picture of the chromosomes within a cell, and it makes sure that you have the correct number. That's its main goal. So to answer your question, the most common cause of Down syndrome, but not always, is an extra copy of chromosome 21. Great majority are caused by this. So I don't think there's a one-size-fits-all answer to your question, and I think it's a personal choice, but would having a confirmed diagnosis, genetic diagnosis, be beneficial? I typically do recommend confirming it. Did they give a reason why maybe they said?
I guess my question is more along the lines of, since we know she has Down syndrome, he's associating that as the reason for the LGS, but I'm wondering if there's more
Something
reason in other areas besides just the fact that she has Down syndrome. Because we had the amnio done when I was pregnant and got the diagnosis pre-birth, so we've done no testing since she's been born,
Okay,
which was almost 19 years ago, so.
yeah.
Unless there's a compelling reason to think that there's something additional going on, like the seizure pattern is not typical for, like, I mean, we definitely can see LGS in people with Down syndrome, like that's one of the genetic mechanisms that can, lead to LGS ultimately. So, I mean, we have, can everybody hear me actually? I will just say, on occasion, we have had individuals who've come to our program who have a genetic diagnosis, and we opt to do additional testing because we don't think that genetic diagnosis fully explains what we're seeing in that patient, right? That they're so not typical that we think maybe there's something else going on there, and it turns out they have two different genetic diagnoses, but that's not the norm, right? It's not common. So I would say, unless your care providers are telling you, well, we don't really understand why this, that, or the other thing are happening, because it doesn't really fit with the Down syndrome diagnosis, I don't know that I would go out and pursue a whole nother round of genetic testing, because it sounds like the trisomy 21 diagnosis was made prenatally. Yeah. Does that answer your question? Okay.
So on a karyotype, one of the things you can find is aneuploidy, an extra copy of an entire chromosome. And then you can also find what we call copy number variants of a certain size. So that would be extra genetic material, so a duplication that you can see here. So this portion of the chromosome has been duplicated. And then you can also see a deletion, so the opposite. And this is an example where the CDKL5 gene has been deleted or taken away. And CDKL5 can also be related to epilepsy and seizures. When it comes to deletions and duplications on chromosomes, it could be isolated to a single gene. However, they're typically larger. And all of these genes are tightly packed on top of each other. So a deletion or a duplication typically includes more than one gene. So they could be at risk for other symptoms related to those genes as well. Another test that might be recommended or done is called a chromosomal microarray, or a CMA for short. And what this does is it looks for any smaller extra or missing pieces or deletions or duplications that might not be able to be seen on a karyotype. So it is fluorescent technology on the back end that is done, and depending on the color that is shown in the region, it tells us if it's the expected amount of genetic material in that region, if there's extra for a duplication, or if there's some missing for a deletion. And then a refresher, so that was on the chromosomal level, what we can do. On the gene level, so zooming in and looking at all of the letters, there are a couple tests we can do. Right now, there's about 20,000 known genes in the human body. And as Beth mentioned, there's about over 1,000 associated with epilepsy and seizures. So as I alluded to in the past, it could just be the result of a single gene deletion. So that entire gene is now missing, and then the protein it should be making is no longer there. This is an example of KCNQ2. But we can also do sequencing. So this is going letter by letter through the gene and seeing if just a single letter change could also be contributing. And this technology of sequencing can also pick up certain copy number variants of a certain size as well. So if there is a large stretch of letters that are missing, they would be able to catch that. So this is an example in the gene CHD2, where typically we're seeing the letter A, we're seeing the letter C, and now one of the building blocks of the protein has been changed, and that is what is leading to symptoms in this individual. Does anyone know if they've ever undergone, or if their loved one with LGS has ever gone an epilepsy gene panel? Okay, about 3 or 4. So this was more common in the past. It's still a good option, but it's picking a set of genes, typically hundreds, that if there are certain types of changes, would be the cause for epilepsy. So it's a predetermined list of genes. And most commonly, it's tens to hundreds. There are some that are in the thousands. And it would detect any single letter changes within the gene, as well as those larger missing chunks, deletions, or extra duplications. Something that's more common now is whole exome sequencing or exome sequencing. They are the same test. So just because the word whole is not in front of it doesn't make it any different. It's just kind of personal preference of what the lab and individual call it. And what this test aims to do is actually look at all of the genes that are known, but it's focusing on those associated with the symptoms that are reported to the lab. So essentially there is a unique gene panel created for every individual undergoing exome sequencing based on the symptoms reported. And that's what we call a phenotype-driven test, so a symptom-driven test. And the main thing with this test is we're looking at all the genes, but within the genes, we're looking at these regions that are shaded in blue here that we call exons. That's how they got the name exome. And we look at these regions because they go on to make the proteins. I alluded to this earlier where we cut out these white or unshaded introns in the process. This looks at about 2 to 3% of the genetic material. And then in the research setting, and there are a couple insurance companies that now will cover whole genome sequencing or genome sequencing. So the aim of this test is to look at all of the known genes and their surrounding DNA, so those introns or spaces in between, because changes there can also lead to symptoms in individuals. This is the one that looks at about 99% of all of the genetic material. About 2 to 3%. And this is also a symptom-driven test, so again, going to look for a potential explanation for the symptoms that are reported to the lab. So again, kind of taking a step back and trying to use an analogy, the genome would be looking at the entire instruction manual, all of the letters, all of the white spaces, and make sure it's like formatted correctly as well. Exome is really focusing on just the regions of that instruction manual that are maybe telling you just how to put the screws in or something like that. And then we kind of went into this a little bit earlier, but we could have the mitochondrial genome sequencing. So that's taking the mitochondrial DNA, so looking at the genetic material from the mitochondria in the cell, again, the portion that comes from the egg, so from our biological mother. We can get the same types of results, but we also get a percentage when we do this. So you can see that every cell has multiple copies of mitochondria. So if we get that a low percentage of them have a genetic change, it's probably not contributing to someone's symptoms. But if almost all or a good majority of the mitochondria, like 80% or more, have that genetic change, then it could be contributing to the symptoms that we're seeing in an individual. I actually want to break there before we talk about potential results to ask if anyone has any questions in general about the types of tests. We'll do more specific at the end, but I do think there's one over here.
So my son, he's been diagnosed since he was, so it's been 11 years. So he started out with that broad one that was just the specific for the epilepsies.
Yep.
And then they did the exome, and then they did the genome. So
Yep.
on the exome, everything came back okay. When he didn't jump to the genome, he had 2 variants that they say are not specific to epilepsy at this time, is how they put it.
Yep.
So that's been about 4 years. How often should we re-look at that? Because
Yeah.
there's been I mean, is it significant to get it? I mean, they said they wouldn't even have to really
Yep.
do anything again, just rerun everything. Is that significant to do that again?
Yeah, really good question. As I alluded to, there's 20,000 known genes right now. And then the ones known to be associated with epilepsy are always changing as we're doing more research and testing more individuals with those symptoms. And Beth also talked about this, how we're just discovering more all of the time. So what you're talking about, not needing a new sample, is what we call reanalysis. With the new information, with more time, more research, can we give an exact answer or clarify the significance of the variants? We typically recommend waiting about 2 years before seeing if there has been a change to the classification of those variants. Most labs, it depends on the lab, will give you an update. We'll reach out to the clinician who ordered the testing and let you know if there's been a change. Not all labs, though. Then you would need to do a reanalysis, which is prompted by either the clinician or the parent of the individual who underwent the testing. Given that your clinician has decided that they maybe aren't, it sounds like the explanation for what's going on, there's no rush or urgency, but you could just take a fresh look at the genetic information and see if you could find an explanation unrelated to those variants as well.
Yes, because he's also having some autoimmune type issues too. He has some EOE.
Yep.
A lot of his things really present. He did really good with IVIG for a while, so they're thinking he has some autoimmune type.
Yeah.
And I mean, they've sent his CSF different places and everything.
Yeah.
So, is there something? So when they look at that exome, if it was something autoimmune, would that show up? Could that possibly show up?
Good question. I'll give kind of 2 answers. So one, you can, when you request a reanalysis, you can add additional clinical information. So if he's had new symptoms develop in the meantime, the clinician should definitely report that to the lab. So that might add genes to the list that weren't included before. Clinical information, sorry. So updated reports or notes from your providers. And then the other thing to consider is, could there be one gene that's explaining everything? So it could be the epilepsy and the autoimmune, then that would have been picked up already if they had just reported epilepsy out.
Okay.
Very good question.
Thank you so much.
Yeah. So that actually is kind of a perfect segue into the types of results we might actually get from testing. So we talked about how we're all very similar, but we have many, like thousands of variants between us. And so we have to determine, are those variants significant or not? So every variant starts out as uncertain, and many of you said that you had a report with an uncertain variant on it. And then it's up to the lab and the clinician to determine, could these variants be downgraded to benign, so just a harmless thing running in the family, you know, contributing to human variation, or is that variant upgraded to pathogenic, leading to a specific set of symptoms in an individual. And many different types of information are taken into consideration. So the individual that we're testing right then and there, if anyone else has ever been seen with that specific variant, if other people in the population that are also healthy have ever had that variant, and then there are some computer modeling that will go into it as well. But it's important to note that when we do testing and we get a variant, nearly 50% or half of them are currently classified as variants of uncertain significance. And that is a large portion, and we are working to try and reduce that, but it leaves many families in this uncertain area. So when we do testing, we could get a negative result. We didn't find one single underlying genetic explanation. We could get an uncertain result, that variant of uncertain significance. We don't know what its effect is. Or we could get a positive or diagnostic result. We did find one single underlying genetic explanation. And if I remember, that was very few families who raised their hands for that. I alluded to this earlier, and this is why we kind of wanted to just go into the nitty-gritty details in the beginning. There are different types of variants that you might see on a report. So there are actually some letter changes that don't change the protein at all. So we call those silent, and those typically are not harmful. We can have missense variants. These are single letter changes that just change one of the building blocks of the protein. Those can be harmful even though they are one single change, or they may not. And then we can have a nonsense. So what this does is it actually stops the protein from being made in its entirety. So the rest of the protein after this area will not be made. And then we can have frame shift mutations. So what that does is I talked about how we read through in segments of 3. So if we insert a letter or take away a letter that is not in a set of 3, we are going to shift every single building block or amino acid, and it's going to change the rest of the protein. It's going to be a completely different protein after that. And although they're all different types of variants, we try and classify them into either loss of function or gain of function. So loss of function means the variant leads to decreased protein function or none at all. And then gain of function, that means that that variant is actually causing the protein to be made more, so it's increasing its function, or that protein actually gains a new function that it didn't have before. So this is actually a really active area of research in the precision treatment. So an example is that if we have a loss of function variant where it's leading to a decrease or absent protein function, which we're seeing here, we might try and switch it or introduce something that would then essentially be gain of function. So we're trying to increase the copy of the gene that is working as expected, since we do have 2 copies of every gene in our body. Any questions there before we go through some testing logistics? So just some things to consider when going through testing. I think there's one up front.
I don't know if this may be sound a dumb question, but is there a checklist for us that we're a newly diagnosed family that I can go through with our genetic counselor to see what he's had done already and what other options we still have left to do, especially now because I think some of his genetic testing was only when he was diagnosed epileptic, and now that he has an LGS diagnosis, if there's something else that, like you said, I have to request for additional clinical information to review, or if there's new testing that he should be getting done.
Yeah, I don't believe there is a checklist. Yeah, I don't know why. As you're saying it, that's a very simple thing. I would say up to this point, maybe things were just changing so rapidly, no one ever kept one up, if that makes sense, right? We only had a couple tests to begin with, so we didn't need a checklist, and now the list is quite long. So definitely something we could look into. There are some good resources. I think they're from the NIH, or it's Medline Plus, which is an NIH website, that will just list every genetic test kind of in a list. However, that's not going to be specific to the indication, right? It's not specific to epilepsy per se, but that could be a start in the meantime while we try and work on one specific to epilepsy. Yes, so that was going to be my next point. Yes, so you should not have to do any work in collecting the clinical information. That should be sent by the ordering provider. The time you might want to bring it up is when reanalysis is going to happen. Say, what symptoms did we report to the lab last time? And are they the same as the current symptoms that are happening now? So again, we recommend about 2 years between the reanalysis. So if it's been about 2 years, maybe now's a good time. You could also bring it up as, is there any other genetic testing options given the symptoms that we're currently seeing right now? It's really a conversation with your team to determine what's best for an individual. Does that answer? Okay. Yep.
Hi, is it possible when you recognize and you isolate the gene that is the problem, is there any way to replace that gene and put the correct one in place?
Very fair question. Unfortunately, right now, for most conditions, no. There are a few. Gene therapies is what they're called. And that is what the research is focusing on right now. And they're in the clinical trial phase for most. I would say having the genetic diagnosis is what gets you into those clinical trials. And having advocacy groups and research into those is what kind of gets the companies to do research into those genes to make those targeted treatments. But that is a very good question, and we were talking earlier that that's where we're hoping all of this is going.
A question I encounter often from patients that we sort of hear is something along the lines of, my insurance might not cover this testing, it's costly and it's out of pocket. Is this something that I should pursue to find an answer for my son or daughter?
Yeah, and I believe Beth will go into a little bit more detail, but exome sequencing is pretty widely covered right now by most insurance companies. Chromosomal microarray as well should be pretty widely covered, particularly for epilepsy and developmental delay. Genome sequencing, there's only 3 or 4 insurance companies that cover it, so otherwise you would want to look for research opportunities. Big academic centers should have them. We have one. I know a lot of partners that we work with have them as well. And then I will say there's a lot of labs, like the genetic testing labs that do the testing, either have programs to help reduce the cost, or sponsored testing, so they work with a company who pays for it, so the family doesn't have to. And then if you do have I almost called it Mass Health, but Medicaid or Medicare, a lot of labs have really good agreements with those types of insurances to reduce the cost to almost $0 for most families, if not $0. I'm just going to quickly go over logistics so that I can pass it back over. So some things to consider, which I know I talked to a family earlier where in the past they had to do a deep muscle biopsy for testing. That is still required in some cases, but actually most times testing can be done through a cheek swab. So it's a buccal kit is what we call it, sent directly to your home, and that's what we're trying to show you here. Other options would be a blood draw or a saliva collection. And then there is the skin and muscle. Those are now reserved for more specific types of conditions where we need to get the very specific type of sample. And then sometimes a urine sample is needed for that mitochondrial testing if we're trying to confirm something. And then with that exome and genome, and even some panels now, we typically ask for samples from biological family members, typically biological mom and biological dad. We have so many variants that make us unique that knowing if something's being passed on in a family or not can be really helpful to the interpretation, and knowing if parents are affected or unaffected. So is the child the first person who's affected and having that variant? And then testing can be completed in as little as a week, and that's typically inpatient in an urgent setting. So for most people, it takes about 1 to 2 months to go through the entire process. And then the chance of getting a diagnostic result, a positive result, really relies on what test is being ordered and the specific patient's history and clinical information. So that's why we kind of emphasize that. And then it is possible that different labs can classify the same variant differently based on internal information. So that's something a clinician can look into as well. There are databases where all labs are supposed to submit their variants to. So that is something to consider when talking with your team as well. So one might classify it as uncertain, one might classify it as likely pathogenic or likely benign if they've seen it in other individuals that they have tested that another lab hasn't tested before. Okay, any questions about that before as I bring Beth up?
I actually do have a
Yeah.
question. Is there a database? My son does have a definitive mutation in the IQ-SEC2 gene, and rarely he's doing very, very well with seizure control. And I'd actually like somebody to know that,
Yeah.
because possibly the medication cocktail that he's on might be specific to his gene. So I just don't know who I would report that to, or is there a database somewhere I can report that? Great.
It's a
Thanks.
perfect
Okay.
segue, yes.
Anybody else had the questions?
Up front. and
Up
then
front,
we'll-
okay.
Since we're a newly diagnosed family, does it matter the genetic testing that he's had in the past, time-wise, like with the medications and CBD he was on? Does that affect the testing because like he had just started the medication versus if he had a repeat, now he's been on the same medication longer? Will that affect the genetic testing with CBD and with his seizure medication?
Good question. Genetic material is determined at conception. So when you swab it and send it in, or however you sent in the sample, it's not going to change from that point, and no medication should alter that.
Okay. Or CBD?
Correct.
Okay.
Okay, so we are going to move on a bit just to selecting tests and, which test is best. Really kind of depends on the circumstance and what tests an individual has had previously. But I do want to just kind of start by reminding all of you about what we know about the genetics of Lennox-Gastaut specifically, which kind of mirrors what I showed you early on in terms of gene discovery, and that is that there's no single gene that is the LGS gene, right? Like we know with Down syndrome, it's always the 21st chromosome, right? With LGS, there's more than 50 different genes to date, and I'm sure that list will grow, that have been identified, variants in 50 different genes and counting in different individuals with Lennox-Gastaut. So this is what we call genetic heterogeneity, that you can have people with a different genetic variant, but who all present similarly in terms of their clinical symptoms. And we know that some individuals have single gene variants, like a single nucleotide that's changed, but we also know there's some individuals with LGS who have larger chromosomal deletions or duplications that involve multiple genes. And we also know that in most instances, the genetic variant identified was not inherited from a parent, but typically arose new or de novo in the egg or the sperm that went to make that particular child. And then down at the bottom is just a list of some of the genes that we know of associated with LGS. It's by no means an exhaustive list, and it changes on a regular basis. So given this, right, how do you figure out what test are you going to do for a child with a new diagnosis of LGS? Where are you going to start, right? And the answer to that, back when I was in graduate school, would have been do a chromosome study, right? Do a karyotype, look at their chromosomes, and maybe you'd find a large deletion or duplication, but most likely you'd find nothing. And if it were 10 years ago, it would have been do an epilepsy panel, right? And if the gene happened to be on the panel that the lab you use includes, then you'd find an answer. If the gene hadn't been yet identified, then you would get a negative result, right? A normal result. I would say at this point, a gold standard for us that we can get covered routinely by insurance is exome sequencing. But if we can get a genome covered, ideally that's where we'd like to start, because that's kind of the biggest bang for your buck in terms of making diagnoses. But to kind of arrive at what our current practice guidelines, I just want to take you really quickly through a systematic evidence review that I conducted with some colleagues. This was on behalf of our professional organization as genetic counselors called the National Society of Genetic Counselors, and we wanted to look across all of the literature to see what was the likelihood of a positive or a diagnostic result for genetic testing, any genetic testing in individuals with epilepsy. And when we first had our medical librarian run the search, there were so many articles that it wasn't practical for us to read them all. So we had to kind of pare it down. And I don't know if you can read this, but the top circle up there, we ended up screening about 6,000 articles, which is still an awful lot. And, you know, we had very narrow criteria in the end out of necessity, and we ended up looking at 154-ish studies, primarily chromosome microarray, exome sequencing, genome sequencing, and then multi-gene panels. And these were papers that were published by some of the diagnostic labs, by research groups, by clinical groups in different medical centers around the world. And ultimately, here's what we found. The highest yield, or the highest likelihood of a positive diagnosis, came from genome sequencing. Exome sequencing was about half that, about 24%. Multi-gene panels, the yield or the diagnostic rate varied depending on how many genes were on the panel. So as you might expect, more genes on the panel, you're going to have a higher diagnostic rate, right? About 19, 20%. Fewer than that, a lower diagnostic rate, around the same as what you find with the chromosome microarray. So basically what this means is if you have 100 individuals who have early onset epilepsy with some developmental features, and you do genome sequencing, you're probably going to make a diagnosis in about half of them. If you did exome, you'd make a diagnosis in about a quarter of them, and so on and so forth. So that led us to put together some professional practice guidelines. So these come from the National Society of Genetic Counselors, but they've been endorsed by the American Epilepsy Society. And there's two recommendations, basically. The first is that all individuals with unexplained epilepsy, regardless of the age of onset of the seizures or the age of the individual at the time of testing, they really ought to be offered genetic testing. Because you sometimes hear about, in particular adult settings, where there's clinicians who don't think there's any point in doing genetic testing because the individual's already 30 or what have you, and, you know, what difference is it going to make anyway? You know, we don't agree with that perspective. So really, any age of onset, any age of the patient, and the testing really should begin with something comprehensive and broad-based. So the days of doing single gene testing are really pretty much over, unless it's a situation where you're really close to being sure that there's a single gene epilepsy, that like this individual has a diagnosis of Dravet syndrome, and you can sequence the SCN1A gene, and you've got something like a 90% chance of making a diagnosis just by doing that. The vast majority of the time, really, you need to start with either a panel or exome or genome, and we conditionally recommended exome or genome over a panel because when you get the parental samples at the same time, you can really reduce the number of variants of uncertain significance. You saw in Jillian's talk, it's about 50% VUSs, right? That is definitely going to go down. I will say that this type of dilemma happened about a decade ago in cancer genetic testing, where there were so many VUSs and it was so difficult to interpret. It's gotten better over time as we've learned more and more, and that will certainly happen. Most VUSs are ultimately downgraded to benign over time, but it really helps to have the trio testing if biological parents are available. It's not required, so it's not if you have a child who's adopted or a child where the other biological parent isn't available or not involved. It's not a necessity for the lab, but it does help with interpretation. And then the second recommendation we made is, just that the test should really be selected and ordered by somebody who is qualified, knows about genetic testing, and there should be pre and post-test genetic counseling made available, which I understand that probably wasn't the case for many of you, which I think is partly an access issue and partly speaks to just how prevalent genetic testing has become in, many different specialty areas, not just neurology. Okay, so let's talk a little bit about access. First of all, we're going to talk about paying for genetic testing, which I think we touched on mostly, but there is quite a bit of variability in terms of payment policies. So it's really important to check with your insurance provider about what they will cover and under what circumstances. Policies change on a regular basis. We are finding that most insurance payers will cover exome as long as you meet the criteria. Not as many will cover genome. Oddly, many will no longer cover panels. And most will cover microarray, as was mentioned before. Usually you're going to need some sort of prior authorization from your insurance company, but that should be something that your healthcare provider takes care of for you. You shouldn't be asked to, call your insurance company to set up the prior authorization, because usually an insurance company needs a letter of medical necessity and some clinical notes. You might still have copays and deductibles, and some insurance companies will require that you have pre-test counseling with a genetic counselor or medical geneticist in order to get approval for an exome or a genome, but by no means all. And then, as was mentioned before, there are some free testing options. Sometimes they come with a little bit of a string attached because they're funded by pharmaceutical companies, and the pharmaceutical company is going to get anonymous data back from the lab as long as you agree to it. But that is a good option for testing that can otherwise be several thousands of dollars if there's no way to get it through insurance. But as was mentioned before, there are some labs, including one that's here in the exhibit hall, GeneDx, that have rolled out free or reduced priced testing, so that's worth looking at as well. And then there's a couple of national research projects like the Undiagnosed Disease Network and the Rare Genomes Project, and also at Children's Hospital, where you can get exome or genome sequencing on a research basis by enrolling in a research study. The pace tends to be much slower. It's not going to be 1 or 2 months. It might be 6 months. It might be a year. It might be 4 years. And research studies by law cannot release results until the finding has been confirmed in a licensed clinical lab. So I know a lot of families get frustrated because they feel like they enrolled in a research study and they haven't heard anything back. And I can speak for Children's. If you haven't heard anything back, it means we haven't found anything. If we find something, we confirm it, and then we reconnect with the family and give them the results. Happy to answer more questions about that process as well. Okay, so accessing genetic counselors, also a little tricky. There are more of us than there used to be, but there's still only about 6,000 or 7,000 genetic counselors in the United States. There's not many in other countries. A reasonable number in Canada, but there's really not very many who specialize in neurogenetics, let alone epilepsy genetics. There's probably a few dozen whose focus really is on the genetics of epilepsy, and probably 7 or 8 of them are at Boston Children's Hospital, so we are not very well dispersed around the country. So, availability can be limited and wait times can be long, even for people who live in Boston and want to come see us. We do have kind of an informal group of genetic counselors that we call EpiGC. We're not a formal nonprofit. Our website is a bit defunct. But we, if you were to say, reach out to me, and you'll get our contact information at the end and say, hey, I live in California, or I live in New Jersey, like, is there someone near me who knows a lot about the genetics of epilepsy who I could go see? Chances are we'd be able to give you the name of somebody. But short of that, there are also quite a number of companies that have popped up in recent years that offer virtual genetic counseling, either through your insurance or for a small fee. They won't be well-versed in epilepsy genetics necessarily, but they can certainly walk you through the testing process and help facilitate at least the testing. And then once you get a result back, you could go to a center that has more expertise. So a couple ways to find genetic counselors are through the National Society of Genetic Counselors, or if you happen to be from Canada, there's a counterpart organization there. Okay, let me just take a short break there before we go on to some other considerations. Right behind you.
Okay, so my daughter is 24. She has a brain migration defect, which is what has always been, this is what's causing her epilepsy. She was tested 24 years ago. Is there a possibility that she has a lot of weird diagnoses and stuff that, nothing really always meshes with the other? Would it be something that we should consider? And does there have to be something new for insurance to cover it? Or would the fact that it hasn't been, it's been 24 years since she's had it? Do you know what
If her testing was that long ago, she hasn't had any of the tests that we've talked about, other than maybe a single gene test or two and a karyotype. Because microarray, what didn't come about until 2007, panels not until 2012, exome was around 2015, and genome is really just, within the last couple of years. So absolutely, I would go back and start the testing. It's kind of like, mobile phones, right? In 2000, I didn't have a mobile phone.
Right.
Then I had like a little flip phone. Then I had a BlackBerry. Now I have an iPhone, and I'm on like version 18 of my iPhone, right? So honestly, it's the same with genetic tests. They are constantly changing, and it doesn't sound like she's had anything in the modern era. And
Right.
I'll be honest, I can't tell you the number of people in that situation where, I've seen people in their 40s and 50s come in and, boom, we've got an answer right away.
So because like I said, like everyone's like, oh, well, because she has this, that's what's causing it. Is it always just a single cause, or could there be something else? Like I know you said if there was a deletion here, that could cause, and then that might make us not give her a certain medicine.
Yeah. My question there would be, what's the underlying reason for the brain migration disorder, right? Because, when you see those little pie charts about the causes of epilepsy and you see, oh, it's structural, oh, it's metabolic. Well, those are all genetic at the end of the day, right? Structural brain malformations frequently are due to an underlying gene variant.
And I was told hers was just a spontaneous one.
And it might, I'm not looking at her
Oh, no, I understand.
MRI or records, right? And that could very well be the case, but, so much has changed. I would revisit it with fresh eyes,
Okay.
I guess is what I would say.
All right, thank you.
Yep.
Getting in your steps. So our hospital, our epilepsy center, we found out by chance that they offer whole genome through a special program, but you have to convince them that you are failing, that you are not doing well, that your disease is progressing, and that you are at risk, and they will pay for your whole genome.
Yep.
That they will cover it then. So they do not offer it otherwise.
Yep. No,
So it's worth asking. It's worth putting it out there, worth asking that question. Because I did it by accident. I said, look, he's falling apart. What is our next step? And she goes, oh, thank God you said that. Because now I can say to you, but
there
I had to ask it.
Vast differences. And,
Right.
At Children's, we have a mission, right, that involves, we're going to make it possible for anybody, regardless of their insurance type or whether they're inpatient or outpatient, right? We're very fortunate that way. But there's some hospitals that won't let their clinicians order genetic testing, right? Because it's a money loser. Or they won't let you do it inpatient because they're not going to recoup the cost. Yeah, right. And often in adult, there's sort of a bias of, oh, why do we need to do testing? Because the only information you're going to take from it is, pregnancy planning, and you're done having kids, and it's sort of it not recognizing all the other, reasons why a genetic test could be important. So there is a lot of variability around the U.S. and around the world in terms of access. So if you happen to be in a place where you're not getting access, that's when I would say, reach out to the NSGC, reach out to our center, enroll in one of the national studies, contact GeneDx to see if you can get it done, through one of their reduced cost or sponsored testing programs, because it's not equitably distributed. Yep.
Earlier I heard that families can have generations of benign mutations. What is the gold standard, the source of the perfect human, and who holds that entity? What entity controls that you're comparing every test against?
So there is no gold standard, right? Which you probably, but there's several databases that have sort of human population sequencing data, typically generated by people who have volunteered their DNA to science, or they've enrolled in some other research study. Like there's a town right near mine in Massachusetts called Framingham, and they did this big Framingham heart study, and all the people who participated in that gave DNA samples, and these are not children, they're adults, they don't have neurodevelopmental conditions, so they have heart conditions, so you wouldn't want to use them as the gold standard frame of reference if you're studying heart disease genetics, but you can potentially utilize that population. But there is no one. There are quite a number of databases that sort of purport to be representative of sort of benign population variability. One problem with them tends to be that they're not very ethnically or racially diverse. So that can be a problem, right? If you're not Caucasian and you're having testing, you might be much more likely, and in fact, in the world of epilepsy genetics, it's already been demonstrated, you're probably more likely to have a VUS result if you're a person of color than if you're Caucasian. So it's a great question. I don't know, did I answer your question?
Yeah, I've assumed different organizations like yours have your standard, but then another group of researchers has a different standard.
Yeah, I mean, these are all publicly available databases. Some of them are from, kind of big think tank research organizations like the Broad Institute, which is in the Boston area. But at the end of the day, I guess, just getting back to Jillian's conversation about variant interpretation, it is a science, right? There are criteria that are used that are set forth by the American College of Medical Genetics that all labs, or at least in the US, are supposed to follow, and many labs overseas do. But it is also a bit of an art at the end of the day, which is why you could submit a sample to 5 different labs, and one would call it a VUS, and one would call it likely pathogenic, and one might call it likely benign, and that's just kind of where we are, because human variability is what it is, and it's probably going to take some time to get to the point where there's enough data, right, to be able to say definitively about some of these variants.
Okay, thank you.
Yep. Okay. All right, it's 2:23. I'm more than happy to run over, and I know Jillian will be as well, but why don't I try to, get us through a few more important concepts, and then I'm happy to stay for more questions. But just a few things to keep in mind if you do go through the testing process. And this one's actually really important. I was talking with someone this morning. There's something called the 21st Century Cures Act. You may be familiar with it, you may not be, but you are required to be given access to your own medical record, your child's medical record, whether it's electronic or on paper. You have the right to see the actual genetic test reports. I'm going to focus on that for now, not the rest of your electronic health record. But if you want to see your report and you ask for it, you should be getting it. Now, I will say at Children's, we just switched over to a new electronic health system that enables people to see the reports right away in real time. As soon as we get them back from the lab, they're in the health record and the parents can see them. That's a bit of a double-edged sword, right? Because sometimes the parent might log in at 9 o'clock on a Friday night and there's this report and they are not written for lay people. And so sometimes it can lead to some pretty panicky thoughts and feelings if you start Googling things. So I usually suggest that people don't look at the report until they've had a chance to talk with us first, so we can let them know how we're interpreting it. But you absolutely should get copies of any genetic test reports that your child or loved one has had, so that you can keep track of, okay, well, you know, she had exome sequencing, but it's been 4 years, so I'm going to ask about reanalysis, right? Or, oh, she only had a panel, and it was 2013, and there were 10 genes on it, and I just came back from this talk and heard that there's all these other options, right? So if you don't know what your loved one has had, it makes it more difficult, I think, to advocate, or you're sort of depending on your care provider to be keeping up with all of the changes, right? Second thing is that when you do broad-based exome or genome sequencing, there's the possibility of what we call secondary findings. Now, I don't think any of you probably are pursuing or have pursued genetic testing because you wanted to hear about cancer risk or heart disease risk, but you can potentially learn these kinds of things when you do this sort of sequencing, and if you're giving samples as parents, you might learn these kinds of things about yourselves as well. So typically labs will have an opt-in or opt-out, so you're not forced to get this information, but it's available to you. Your child, if that's who your loved one is sequenced, and there are typically a set list of genes that the lab will look at. And again, this list is compiled by the American College of Medical Genetics. It changes periodically. There's 80 plus genes on the list right now. They are all for adult onset conditions that are considered to be medically actionable, right? So the idea is there's something you can do to intervene or a prophylactic measure. So it's just a choice to be made at the time of testing. And whoever's ordering the testing for you should be asking you, do you want this? Do you not want this, right? So that there's no surprises. That issue sometimes brings up questions about genetic discrimination. Like, oh, what if we have testing and I find out that I have a breast cancer, variant that predisposes me? And so we talk sometimes about genetic discrimination, and I will say there are not a whole lot of examples, at least in the US, of actual discrimination based on genetics. The worry is real, and it's there. There are some federal protections, so this is called the Genetic Information Non-Discrimination Act, or GINA, but there are some things that this doesn't cover. So GINA pertains to health insurance and employment discrimination. It has no benefit when you're thinking about long-term care insurance or life insurance, and there's certain employee groups that are not protected by GINA, ironically enough, like federal employees, but also small employers. So there are some loopholes there, and in some states there's additional protections. I know there are in Massachusetts, but it's something just to think about, before you embark on genetic testing, especially if you're doing trio testing. Do you have your insurance policies in place if you're going to go down that road, before you decide to get back secondary findings, for example? Okay, in terms of resources, so this kind of gets back to the question of where do I put my data, right? There are several organizations. So the National Brain Gene Registry is one of them, and then the Simons Searchlight Project, which are really specifically focused on genes that play a role in neurodevelopmental disorders, including epilepsy. And I'm almost 100% certain that that IQ SEC2 gene is on the brain gene registry list. So if you end up with a genetic diagnosis and you're kind of wondering, okay, where do I go with this? How do I connect with other people? There may be an advocacy organization that's specific to that gene that you can connect with. But you can also go and deposit information into the brain gene registry. You can go to something called MyGene2 or Genome Connect, and you can put in the gene and the variant. Some of it's anonymous options, some of it's not. Like if you want to be able to connect with other people, it kind of depends what you're hoping to get out of it. And then there's also something called the Rare Epilepsy Network, which is wonderful for a quick look to see. Like if you've been Googling and you can't find your gene and there's not even a Facebook group, you can go to the Rare Epilepsy Network and see, do they have a gene-specific group listed on their website? Because they kind of keep track of them all, or in a major clearinghouse for all the genetic epilepsy organizations. Okay, let's talk a little bit about interpreting reports in like 2 minutes, because I know we're at time. Maybe we'll just take you through one or two of these, but a couple things just to keep in mind when you look at reports. You're going to get the gene name, you're going to get details about the variant and how it's classified. You're going to also find out about the inheritance pattern, right? The two most common being dominant and recessive, right? So dominant inheritance, typically you would see family history, individuals in multiple generations all affected with the same condition. That's typically not what we're talking about when we're talking about genetic epilepsies. Recessive inheritance, by contrast, there are often lots of people in the family tree who are carriers, who are not affected with symptoms, but if two carriers have children together, there's a 1 in 4, 25% chance in any pregnancy for that child to inherit both variants, right? One on each copy of the chromosome, and then actually be affected. So recessive inheritance would be something like sickle cell disease or cystic fibrosis or Tay-Sachs, or if you're familiar with those. Dominant inheritance, something like an adult onset Huntington disease would be an example. There's lots of other conditions that follow dominant inheritance patterns. And then there's other inheritance patterns like X-linked, and which I won't go into great detail about, but just to give you a sense of the inheritance pattern can matter when sort of interpreting whether the variant is relevant or not for your family member. Okay, so here's an example of a result from a lab. I actually can't really see it from over here, but so you've got the gene name all the way over to the left and the inheritance pattern, which is dominant. So you're starting to think, oh, okay, it's dominant, it runs in the family, right? But if you look in the inheritance column, you can see that it arose de novo, or new, which means it wasn't present in either parent. So this individual with this de novo variant, if they had children, there'd be a 50-50 chance for them to pass the variant on. But this individual's parents likely don't have the variant because it wasn't identified when they were tested, if it was trio testing. The rest of the information that you see in the middle section there is really information about the variant. So the variant column is sort of the address, right? So it's the specific nucleotide codon, that triplet letter code that Jillian was talking about, and then it's also the amino acid. That's the P part. So the C is the codon, and then the P is the amino acid within the protein. So if you have an amino acid that's, if you have a protein that's 100 amino acids long, it might be the 100th amino acid or the 158th amino acid, and then the amino acid change. And this, I can't read it from over here, but so it tells you what the amino acid change is, and then you get information about the classification. So in this case, a likely pathogenic variant. So if you read the interpretation, though, right, you look at your little pedigree here, you've got the biological parents, and they have 3 children, 2 are not affected, 1 is, so it's de novo, not likely inherited based on testing of the parents, but you have this little disclaimer here about parental mosaicism. And what that is basically, you can have individuals who have mixed cell types, So in parents, usually that's going to be confined to what we call the gonads, right? The egg or sperm cells. So if I, as a parent, have egg cells and 2% of them have a variant in, let's say, the SCN1A gene, then there's a small chance each time I have a child that that egg will go to form that baby and therefore might have SCN1A related epilepsy. But I don't have it because it's only in my egg cells. Does that make sense? Yeah. So it's the one thing to think about if there's a de novo variant, if you're thinking about having more children, is the possibility of mosaicism. It's not really possible to test egg and sperm. You can, as a parent, depending on the gene, oftentimes get something called mosaic carrier testing. So you're essentially telling the lab, I want you to do a deeper dive for the variant you told me I didn't have to see if there's low-level mosaicism. It's a separate test because it's a slightly different testing technology. But I typically suggest that for families who want to have more children if they want, reassurance. But short of that, there is also prenatal diagnosis. That's an option if you know the child's diagnosis, regardless of mosaicism. Okay, and then this is just a recessive example where, both parents, each of them has a variant, and, you can see that individuals who are shaded half white and half black are carriers who don't manifest symptoms, and only the individuals who inherit 2 copies, one from each parent, are going to manifest symptoms. I'm just going to go a couple more examples because I think these are important because it sounds like a lot of you have variants of uncertain significance based on testing. Sometimes you might end up with a variant of uncertain significance because you've done a panel test and the parents haven't been tested, right? So the first step is going to be to do parental testing. If neither parent seems to have the variant and it seems to be de novo, it might be tempting to say, okay, that's the answer. That won't necessarily, from a lab classification perspective, land you in the likely pathogenic or pathogenic range. You might still be in the VUS range based on current classification rules about variants. So that's when you might want to ask for reanalysis, or you might want to consider additional, more comprehensive testing. If the results of the parental testing come back and one of the parents has the variant, the lab's probably more likely to downgrade it to benign if there's no significant family history. And then lastly, I just want to mention candidate genes, because this happens fairly frequently, more than I might have expected. But, so this is a gene called PP2R5E, I think, from over here. And you can see the disease says none currently described, and the mode of inheritance says unknown, and it's inherited from unknown, and it's called a variant of uncertain significance. But it's specifically what we call a candidate gene, because there's no individual, there's no human with any specific medical condition who's been reported in the literature to date. So it's a candidate because of maybe what the lab knows about that gene and the protein that it codes for. It has some important role in the brain or the central nervous system. Or maybe they have internal data. They have 5 individuals who they've tested previously, and they're collecting this data, and they're kind of not ready to publish yet because they don't have enough evidence, but they have good reason to suspect that it's a candidate gene. So technically it's a VUS, but it is also a candidate gene because we don't know a lot about it. And usually what we do is we put that variant into something called Gene Matcher, and we connect with researchers and clinicians all over the world and see how many other individuals are there who've been tested, who have variants in this gene, what's their clinical picture look like, and is it a match? And that's often what will lead to a publication and a new gene discovery. So I would say a candidate gene is not, the end of the road. Oftentimes in our experience, it does end up being a diagnosis. Not always, but often. Okay. And then this is just some information about our program at Boston Children's, the Epilepsy Genetics Program. We have a website, we have a phone number. This is our contact information if you wanted to reach out to us. I think these slides are going to be made available. I'm not sure. This is all being recorded, I believe. So, Tracy Dixon Salazar has our contact information, so I'll give you a couple minutes to write that down. But please don't hesitate to reach out if you have questions or if we can help to facilitate connecting you, with someone who can help you get testing regardless of where you live. So I know we're a few minutes behind, but I will stop there and just see if you have any other questions for us before we wrap up. And thank you, because I know that was a lot.
Hi.
Hi.
So I have 2 boys, ages 6 and 8, and we got genetic testing over 5 years ago. They're going to redo it soon. But my question is, if we have been told by the genetics counselors that there were 12 others with this, none of their characteristics are the same as ours. We were told years after that.
Right.
And we're with the UDN for fruit fly models with no characteristic changes either.
So can I just ask, both boys have the same variant?
Correct, but they're both VUSs, but me and Dad are carriers.
Okay.
So I'm trying to figure out, you did say sometimes it drops down to benign, but in our situation, probably not. We should wait it out? Or what do you recommend?
What's the gene?
APC2.
And so you're saying each of you has a VUS, and both boys have inherited both VUSs?
Correct.
Okay. And you're wanting to know, so are they suggesting that you repeat the testing or that you do additional testing?
They suggested we do the UDN fruit fly model, which we did, but
Right.
they have found nothing, and it's been almost 6 years.
And that's since the testing that identified the other VUSs?
Correct.
Okay.
Tricky.
And the UDN you enrolled in what year?
It was almost 6 years ago.
Almost 6 years. So one thing I might consider doing, do you happen to know what lab did the original testing?
No, not
Okay.
off the top of my head. It's one of the more common ones, but I don't know.
Do you have a copy of the report?
I do.
Do you want to come tomorrow morning and we can just take a peek?
Sure.
Yeah, we're going to be in one of the little rooms, the Ask the Expert rooms, from like 7:30 to 8:30, because it might be easier for me to see the actual report. I mean, sometimes what I will do as the counselor is go back to the lab and say, hey, have you reclassified these variants, right? And that doesn't even require a reanalysis, a formal reanalysis. It's literally just going back and saying, have you reclassified them, or are they still VUSs, right?
Okay.
Right? And then the other question is, does the clinical history fit well, given what we know about individuals with variants in that gene and your boys? And if it doesn't, then we would probably pursue like the next step in testing. So like if it was exome, we'd probably do genome.
It was genome, I do
Yeah.
know.
The original one?
Yes.
Yeah. Yeah.
Okay. And if I can find it or if I can't find it, can I email you?
Yeah.
Okay.
Yeah.
Okay, great. Thank
Okay.
you.
Yep.
I think we're good. Thank you.
All right. Thanks so much, everybody.